So we have our next speaker, Professor Jayashree Saha from Department of Physics, University of Calcutta. So it gives me a pleasure to introduce Professor Saha because in my MA series I attended her classes during 2010 to 2012. So it gives me a pleasure to interact with her in this platform. So thank you very much, madam, for coming here. So Professor Saha did her PhD from and MSc both from Calcutta University. Her research area includes mainly statistical physics and soft quantum matter physics, mainly the transport through biomembrane, transfection of DNA complexes and gene therapy, etc. She has numerous publications and she attended lots of conferences and seminars. Today, she will talk about the theme of our seminar. So with this, I will invite Professor Saha to deliver her lecture. Before that, so our departmental teacher Gautam Bishas will felicitate her with some proclamation. Madam, the stage is all yours. Please proceed. Thank you.
I would like to thank the organizers to give me a chance to present my talk here. So mine is a bit of a formal talk. So I'll try to explain the physics and mathematical part to the biology students. So please stop me if there is any problem with the mathematics and the physical understanding. So the title of the talk is Physics and Cell Biology, Vision of the Future. Mainly, so we are the learners. That is Pranay Pal, Somala Bhomik. Shayantan Mondal is here. He will present his work. And Ovirup Das is also present here. So we worked in Department of Physics, University of Calcutta. So I will first address the question that is, does cell biology need physicists, or it will remain as a captive of chemistry and genetics? Is there any need to apply physical laws based understanding in cell biology? So this is the main topic I will address today. And so this is, you know that this is the typical cartoon of a eukaryotic cell membrane. And let us first see what are the functions of a cell. So cells are the fundamental units of a living system, and it's at the primary level, its functions are basically to grow, replicate and divide. But as human beings, we want more. So we want to guarantee ourselves in a long and healthy existence that is too often thwarted by this disease. So what is disease? Disease is the disruption of normal activity, and it stops the body which it planned to do. So disease, so there are many causes of disease. So that can be toxins or genetic or cellular defects. Those can be inherited or acquired. There can be bacteria which can attack the body, and also the virus, fungus, etc. So these are the causes of disease, and they attack the body at different levels or stage. So what now their action is, so their involvement is in different time and length scales. So I will explain what are these time and length scales are. What are these things? So human body is a multi-level biological organization. So these are the levels. So this is molecular level. Molecular arrangement gives cellular structure. Cells makes tissues. Tissues forms organs. So these are different kinds of length and time scales. So the general idea is that gene-related factors affect the characterized major aspects, but this is not true. Any level can affect the other levels. That is, if there is any problem with the molecular arrangement in the molecular arrangement, then it can affect the cells. And if there is any kind of problem in the cell level, it can again disrupt this molecular arrangement. And also these are organic molecules. These molecules can also change their structures. As these are the levels, so this is molecular level, this is cell level, this is tissue level, and this is the whole body level. So human body is the interrelated collection of systems working at many spatial and temporal scales, and this is important. There are many levels or scales. And individual physiome emerges at system level collectively. So in biomedicine, what people usually do that they study the system level, the processes at system level, because one level influences the other equally. Anyway, disease is a cellular level process which involves which can affect molecular organization, cells, even multicellular systems. So diseases are at their heart cellular process mainly, which can affect the organs also. Now to combat with the disease system, what do we have in hand? Recent technological and experimental advancements gave us detailed information about the cell behavior at molecular level. We can now resolve or quantify the processes inside the cell even at the molecular level. But in these works, physics acted as a supporting means, not as a guiding factor. So their physics takes a secondary role, not a primary role, when any biological system or medicine or medicinal subjects or something, their physics always take a secondary role. And in the past, we have seen that cell biologists were extremely capable of making good progress without much need of basic physical understanding or knowing much physics. But now that these high throughput methods, high throughput informations are coming, and so a lot of data are amassed. But when we put them together, it becomes harder and complex if we want to interpret those data. So this is the bottleneck that is for any branch of science like medical science or even soft matter physics or physiology or any kind of biological system, which try to understand the molecular level processes to explain some macroscopic phenomena. Macroscopic means which we can feel or which we can see in the experiment. So this interpretation becomes harder. Now I'll explain the role of physics. So our aim is to study human system biology, applying laws of physics, and to apply it to clinical application. So this translation is very important. It has to be applied to clinical applications. So understanding macroscopic collective behavior collective phenomena from the complex microscopic structure and dynamics. To understand this, we need to make some models. So this physical model should incorporate information at length and time scales ranging from molecular means nanometer, nanosecond range, to clinical levels, that is meter, days to years. Because the human system is a collection of levels at molecular level, at cell level, at organ levels, and then clinical levels. Clinical levels is concerned with this meter scale or days to year scale, whereas molecules belong to this nanometer, nanosecond scales. So for all the levels, this physical model should be different. So physicists have, in the past, physicists are very successful by providing simpler view of astounding complexity of cellular biology. That means some models are made where some fine interaction details, some data are neglected, and the important things are considered. So physicists can do that in other branches of physics also. This is called reductionism, but reductionism is not always useful. But physicists have done very well in determining what details are important and which are not. And cell biology is waiting for these instructions. So we need physics and physical laws to understand the huge data we are collecting from the experiments and technological viewpoint, technological means. So this is the role of physics, and we will take the reductionist view for this kind of thing. These are some examples where molecular motor, cellular movement, and how cells swim or cell division, how cells interact with the environment. These are some successful physical biology. So just first I'll explain what is because of the modeling, physics-based modeling using physical laws in biological system, how we can do the modeling. So this is the structure, chemical structure of a lipid molecule. Lipids are there, are in abundance in human body. So these are the lipid molecular structure. So when it is this kind of molecule are modeled in all atom models, all the atoms are taken, and these two molecules are some kind of interaction are assumed between two molecules, and then it can be studied. Another way is this reductionism, that is a group of atoms are bundled together, that is in the head group. The head group region is here. This is the second kind of model. So this is the head group and this is the tail region. So all the interactions for those atoms were bundled in a single ellipsoid, and it will interact from the center of the ellipsoid. So there are only two force centers in this model, where there are many force centers in this model, in all atom models. So all atom models are not computer efficient. Not only that, for all atom models, what happened, the large number of data, those are not needed. But in this postgrain models are not only economical, but it gives some useful perceptions which any branch of science cannot conceptualize. So there is a elimination of fine interaction details, but understanding basic interaction responsible for certain property. So these are the future models. That is the postgrain models, where the important interactions are only considered, others are just neglected. So it takes on much smaller. They are much economic. That is, they are not very expensive. So this kind of future research work. And so as a case study, how this kind of physical models can be used, physics-based model can be used to sort out an unsolved problem of medical science. I'll just give an example. So our project aim was to develop smart drug or gene carriers. It was just the initial aim, but we landed after some time. We landed to some different objective. And this for that we did some postgrad level modeling. So what is gene therapy? Defective DNA can be corrected by injecting normal genetic materials inside the cells, and this will be rejected by immune system. Now viral vectors, viral have effective mechanism, and they just did this for years and years. That is, they have effective mechanism, so they are very efficient in it. Viral vectors have effective mechanism, but they cause adverse effects. If you use viral vectors, it can give leukemia and so on. So there is another kind of drug delivery system, that is liposomal drug delivery system, that may revolutionize gene therapy. So we tried with that. So this is some, the mechanism, the procedure is like that there is some drug or a part of a DNA that is packaged inside some lipid molecules, because the cell membrane are made of lipid molecules. So if you give the drug or the cargo, that will go to the cell, and there will be some diffusion near the cell level, and the cargo will enter in the cell. So the aim is to break the cell defense. So there should be some membrane fusion. So there should be bilayer destabilization and lipid fusion. First we'll see, this is a cartoon of a cell membrane. So the lipid molecules gives this is the main matrix. So lipid molecules that I have shown earlier. So they make the basic structure. Water comes here, it cannot go inside. Lipid tails are, these are amphiphilic molecules, that means here this part is water loving, and this yellow part is hates water. So this and majority of lipids are phospholipids, and they make a matrix. So this is a part of a cell membrane. And proteins, cholesterols are embedded inside the molecule. So and Sayantan will talk about this cholesterol part. So this is kind of lipid matrix. And lipids are amphiphilic, so they are, because they are negatively charged phosphate group and positively charged nitrogen group. This creates an effective dipole movement. And tails are hydrophobic and non-polar. And these are some lipid phases. So they make self-assembly and so on. And this is the phospholipid bilayer matrix. It is just a bigger picture. One molecule looks like this. So here is one lead group, and there is some zwitterionic interaction that occurs here. And this tail part is, these are not charged. Now our question was, how does biomembrane matrix keep lipid bilayer structure in living cells? Because we have to break this. That is the bilayer structure. Otherwise the thing that we want to introduce inside the cell will not go inside. So we know water has important role, but we cannot take out water, so we search for something else. And what we have seen, that anesthetics, some electrolyte imbalance or alcohol, those are sense regulators. If you give anesthetics to someone, there will be some problem with the senses. If you give alcohol, the same thing occurs. If you give them that much alcohol, And also you know that if there is sodium potassium imbalance, then also there is some problem with the senses. So these are they related? Because then do they change or destabilize? So our question was, do they change or destabilize lipid matrix to cross several membranes including blood-brain barrier? Because in our brain there is a big defense system, otherwise everything will go to the brain. But that also was anesthetics, electrolyte imbalance and alcohol, they can go inside. It can do something in the brain. So central nervous system, they can get access to the central nervous system also. So what happens? Because water gives lipid bilayer, but is there any destabilization in the lipid matrix so they can go in the brain? Then we have collected some experimental results. That is preferred. We have seen that the preferred orientation of PN dipole moment vectors at room temperature is on the average along the plane of the layer. That is, this is the layer plane, and the direction of PN dipole moment is along this direction. There is one criteria, that is Mayer-Overton criteria. It is a debated issue for more than 100 years. Nobody knows why anesthetic works in our brain. So correlation of anesthetic potency with dissolving ability in olive oil is the only thing that one can understand whether this chemical or this thing will act as anesthetics or not. And this is the only thing that anesthetic potency can be measured. But this is a very debated issue. So, but we have seen that main difference between anesthetics and respective non-anesthetic counterpart is that only the anesthetics have oriented dipole moment. That dipole moment, this one is along this direction, so it is perpendicular to this layer, and their accumulation near the head group region changes orientation of the lipid head group dipole. That when you put anesthetics, there is any kind of imbalance in electrolyte imbalance, or you put alcohol, then what happens? The direction of the dipole moment changes. It is going along this direction or some other direction. And the presence of anesthetics and ions changes dipolar angle of lipid head group with bilayer normal and controls ion channel opening or closing. And this is responsible for ion channel opening and closing. So these are all experimental data that we collected. These are done by some in the past. So we take some coarse-grained model and average over averaging over non-essential details for time and length scaling in that region, that is the cell level region. And we replace the group of atoms by larger units, and this is a powerful tool to study much larger systems with minimal loss of accuracy. So just a bit how this modeling can be done. So this is a very well-known potential in physics. That is, this is Lennard-Jones potential. Lennard-Jones potential is when these two atoms come closer, the energy will become minimum, that is the most stable state. The system prefers this position, so this comes closer, and by this way Van der Waals, this is the origin of Van der Waals interaction also. And when the two atoms go further, this will change and the energy will become negative, but it is close to zero. But when they try to penetrate one another, then there will be a hard repulsion, that is there. It will not be allowed because of the electron cloud overlap. So this is the minimum. At this position where this potential is energy is minimum, this is the most stable condition. So this is the Lennard-Jones potential. So but this is applicable for this type of spherical particles. Because there are two things here, two parameters. One is sigma. Sigma means how close they can come, and this epsilon is the measure of this minimum, magnitude of the minimum. So this sigma, that is this, if you come from this side, from that side, from that side, from that side, it will be same. So this is a constant, and this epsilon is a constant. But in our case, the molecules are not spherical. So these are ellipsoidal, and physicists love this Gaussian type of thing. So this is a superposition of two Gaussians, and our postgrad model is like this. There is a molecule which is ellipsoidal, and one head group is there, and along this, and the dipole orientation you can control from this part, in this terminal part, and two molecules can interact in this way. So molecules are ellipsoidal, and this is again a two-side pair potential. So electrostatic interaction in the head group region are taken care of by this very known dipole-dipole interaction term. So two dipole interacts through it. But here the problem is, unlike these spherical particles, these two molecules can come closer if they have this kind of orientation. But for this kind of orientation, the center of mass cannot come just like this, so it is bigger. So this sigma should be a function, and also the epsilon, that is the length energy parameter. This should be functions of these orientations and their distance. So the potential is changed in this way. And these are some, so just this graph. So when the molecules are in this direction, in this configuration, the potential energy is much lower. For here it is like this, here it is like this, so there is a comparison between this, and these are some forms. Now if you do some molecular dynamic simulation, these are some technical points regarding the simulations. But initial configuration is isotropic, and phase change is obtained by decreasing the temperature. So molecules have same dipolar angle and state for each system. So we change the dipolar angle for the molecules, and we started the simulation from a configuration like this. So if you lower the temperature, it is a high temperature phase. If you lower the temperature, you will see that for theta equal to 0, that is dipole angle is along this direction. When it is 45 degree, that is molecule dipolar directions are 45 degree or 60 degree or 90 degree. Then you see that here there is this bilayer arrangement that we can see in a membrane. So for 90 degree, that is naturally the lipid molecules in a cell membrane take this angle, that is theta is equal to 90 degree. At 90 degree, the molecule, there is bilayer, but at some other degrees, no bilayer kind of thing happens. So when you give some anesthetics or some other electrolyte, if you or sodium, potassium or alcohol, then what happens? That is, there is bilayer destabilization. So that things can go directly to the brain, because this means the defense in the system, and this breaks. So in this what happens, that with this simple model, just we can explain why there is a sense problem in the senses when the some other things like anesthetics and etc. are good to a patient, are given to a patient. So this is one example. But the problem with this model is that the molecules have two chains, not a single chain, so you cannot take a uniaxial ellipsoid, and also in the head group region there should be water. In the previous case we did not consider water explicitly. So the next modeling, that is physical modeling for biological systems, what we did, we have taken care of this biaxiality. This is known as biaxiality because molecules are like this. So this A and B are different, and molecules in non-cylindrical symmetry and biaxial ellipsoid represents the tail part. So this kind of thing are taken. Again, there is some modeling, and this modeling I'll talk a little bit about it. So this is a joint distribution function, and I'll skip this thing. So then this is those are for biaxiality, and the tail attraction provides the WC thing, that is a spherical phase. Also we got this is known as vesicle, and if you cut this vesicle you will see inside there is water, outside there is water, and the little head groups, that those are this blue kind of thing. They are in interaction with water. So this is isotopic phase, this is bilayer phase, this is vesicle phase, and this is the cut view. So with the introduction of water, we got this kind of vesicular phase, and we get curved vesicular phase at high water concentration and flat bilayer at lower water concentration, that we can see in the experiments also. So this generalized model is suitable for simulation of systems having mixtures of similar dissimilar molecules and their components having different aspect ratios and symmetries, because symmetries are very important in physics. So we have to take the symmetries. So what are the applications of these models? That is, you can use it in biological system. You can mixture various kind of lipid. Cholesterol is another kind of lipid. So you can mix this cholesterol with lipids. You can study RAPs, and also DNA can be studied with this kind of potential. Colloids and surfactants are also some other systems that you can study with this generalized model. So this model can also be used as mechanical and medical purposes, that is for robotic hand. So drug design, about the drug design kind of thing I have talked, and there are some industrial use. So our future plan, because there are some mathematical details. So we want to make package for molecular dynamic simulation, and we want to translate it for RAP formation, insulin resistance, and so on. So Chandra will talk about this cholesterol kind of thing, and this DNA will be discussed by Obiru. So they will come after me, and so this is the role of physics that we can use in this levels, or in this scales, that is nanometer to micrometer scale, in for temporal and also for length scales. So there are some other models, physical models also for some other levels, but I am not going to discuss it today. So now Chantal will talk, and we will talk about that. Thank you.
অ্যালগরিদমটা কি এবং সিমুলেশনে কিভাবে হেল্প করবে?
এটা হচ্ছে যে মলিকুলার ডাইনামিক সিমুলেশন করতে গেলে ডিফারেনশিয়াল ইকুয়েশন সলভ করতে গেলে তোমাকে একটা টাইম স্টেপ ঠিক করতে হবে, যাতে তুমি যদি অনেক বেশি টাইম স্টেপ যদি তুমি টাইম স্টেপকে অনেক বড় নিয়ে নাও, তারপর তোমার কিছু ইনস্টেবিলিটিস আসে যখন তুমি ক্যালকুলেট করছো ফোর্স অ্যান্ড টর্ক, তাহলে কি হবে সিস্টেমটা যেটা ইকুইলিব্রিয়াম থেকে কনফিগারেশন থেকে ঘুরে চলে যাবে। এই জন্য তোমার কাজ হচ্ছে যেন ওটা ইকুইলিব্রিয়াম কনফিগারেশনের কাছাকাছি থাকে। সেইটা করার জন্য লিপফ্রগ অ্যালগরিদম একটা অ্যালগরিদম যেটাকে এবং এই প্রোগ্রামগুলো সমস্ত আমাদের নিজেদেরই করা, এগুলো কোনো প্যাকেজ ইউজ করা হয়। তার কারণ হচ্ছে এই মডেলিংই এখন এক্সিস্ট করে না, এগুলো আস্তে আস্তে আমরা পাবলিশ করছি। আমি যেটা চেষ্টা করেছি সেটা যে আমাদের যা লিমিটেড রিসোর্স, যেন আমরা ক্যালকাটা ইউনিভার্সিটি থেকে এসেছি, কারণ আমরা ইউনিভার্সিটি আমাদের খুব লিমিটেড রিসোর্সেও কীভাবে একটা বায়োলজিক্যাল সিস্টেম যেটা এত প্যারামিটার আছে এবং যেটা এত কমপ্লিকেটেড, তবু সেটাকে ফিজিক্যাল লস দিয়ে কীভাবে আমরা তার আন্ডারস্ট্যান্ডিংটায় আসতে পারি, যেটা আমরা অ্যানেস্থেটিক্সে বললাম। এক্সপেরিমেন্টগুলো কিন্তু ছিল যে এইভাবে ডাইপোল ওরিয়েন্টেশন চেঞ্জ করে দেয়, কিন্তু কেউ এটা বুঝতে পারেনি যে এই ডাইপোল ওরিয়েন্টেশনটা চেঞ্জ করার জন্য সেল মেমব্রেনে কী হচ্ছে। তো এখানে যেটা দেখা গেল যে সেল মেমব্রেনের যে বাইলার ম্যাট্রিক্স সেটাই নষ্ট হয়ে যাচ্ছে। সেটা যদি নষ্ট হয়ে যায় তাহলে তো ব্রেনে চলেই যাবে। কাজে এইভাবে ব্যাপারটা বোঝা গেল যেটা শুধুমাত্র অলিভ অয়েলের পোটেনশিয়ালি আর দেখা হচ্ছিল, যেটা তো কোনো একটা সাইন্টিফিক তার কোনো নেই, মানে কিছু দেখা গেছে এইভাবে হয় তাই করা হচ্ছে। কাজে এইভাবে আমরা কিন্তু বুঝতে পারবো ফিজিক্যাল লস দিয়ে যে বায়োলজিক্যাল সিস্টেম, যদিও এটা একটা কমপ্লিকেটেড সিস্টেম, ফিজিক্যাল লস নিশ্চয়ই আমরা অনেক যেটা এলিমিনেশন করা হয়, রিডাকশনিজম করা হয়, কিন্তু তা সত্ত্বেও ওটাকে কিছুভাবে যেন ব্যবহার করে কিছু কিছু বায়োলজিক্যাল যে এগুলো আছে যেগুলো কেমিস্ট্রি পারবে না, জেনেটিক্স পারবে না, বায়োলজিস্টরা পারবে না। কেন পারবে না? কারণ তারা রিডাকশনিজম করে না। তারা যত বেশি ডেটা আসবে, যত বেশি আসবে তাদের সেটা সুবিধা। আর ফিজিসিস্টরা কী করে, ওই সব ডেটা থেকে তার একটা সিমেট্রি বা সিমিলারিটি বা এরাও একটা কিছু খুঁজে বার করার চেষ্টা করে, যেটা হয়তো খড়ের গাদায় একটা খোঁজার মতোই, কিন্তু সেইটা যাতে বার করা যায় এইটুকুই আমাদের প্রয়াস, ছোট প্রয়াস। ঠিক আছে, এবং আমাদের বড় কম্পিউটার নেই, কিছু নেই, কিন্তু তার সত্ত্বেও এটা করা।
Thank you, madam, for such an illuminating talk. So I request Professor Parthasarathi Majumdar to give felicitate madam with some token of appreciation. The big round of applause for